Hello and welcome to another edition of James and Chris Science Videos, where today we're going to be looking at measuring the rate of photosynthesis using biomass as our indicator. So the same as with the other videos, when it comes to photosynthesis, I think it's really important to remind ourselves each time of the equation for photosynthesis, because knowing it off by heart is really important. So there are the reactants and the products. In this case, the reactants, the raw materials needed, are carbon dioxide and water. The products, the things that are produced, in this case, are glucose and oxygen. Not to forget that you need the chlorophyll and the photosynthesis as part of the reaction, although they don't take place in the reaction. They're there to speed the reaction up. So we need to start off with, first of all, what is biomass? Well, biomass can be split into two different words. Bio meaning living, and mass meaning the amount of particles or amount of matter that is contained within that object. So basically, all the stuff inside a plant or a human being or any living organism that is part of that living organism. So it's the mass of the organism. So in this case, how do plants gain mass? Well, when you go back to look at the photosynthesis equation, the two reactants is a gas and liquid. The liquid comes up through the roots and the gas comes from the air, and they are used to fix into glucose, and glucose is a solid, and so that's going to remain in place. Now, glucose presents plants with problems because glucose is soluble. It'll dissolve easily in water, and if the plants just made glucose, then they would start losing that glucose into the soil. So they need to find an insoluble way of storing the glucose, and they do this by turning it into starch. Now, if you've done the human as organisms topic, you know that enzymes turn starch into glucose. Well, plants do the opposite. They turn glucose into starch, and that's where it's then stored. We then eat it and we have to break it back down again. So the way of looking for biomass in plants is for looking for the starch. So what we want to do first of all is to prove that starch is in plants, and we're going to look at testing a leaf for starch. So I'm going to transfer you over to the lab in a second where Mr. Kipax is going to show you how to test a leaf for starch. I want you to watch him carefully because afterwards I'm going to be asking you to write down with diagrams the different stages that he does. I'll also be asking you questions about how he approaches this experiment. So, off we go to the lab. Let's go see Mr. Kipax in action.
Right, what we're going to do is show you how you prove that a plant has been undergoing photosynthesis and do the starch test on a leaf. Now, what I've got over here is a plant taken straight from the prep room this morning. Over the years, this plant has given up many, many leaves and is looking a little bit ropey, but we do try and look after it. It's got some nice green, bright leaves here. Looking at it, it suggests that this plant has been carrying out photosynthesis with these wonderful green leaves, some of it in a bad state. What we're going to do is simply to prove that this leaf has taken or has been photosynthesizing and producing starch. I'm going to take off a leaf. Sorry, little plant, but I'm sure you'll grow some more. We'll take our lovely green leaf and we're going to test for the presence of starch within this leaf. If you remember back to previous modules, you should know that in order to test for starch, you use this. Now, before I even tell you what it is, you should be able to say to yourselves, yes, that's nasty stuff, very careful with it. It is iodine, but iodine solution. Always remember, the iodine is dissolved into the water to make a solution of that. Now, if you take iodine solution, you put it onto starch, you should be aware of the colour changes. We'll come back to that when we do it in a minute. But if I take some iodine solution and I just drop it carefully onto the leaf here, this will show me something that's quite magical. Or not. What you notice is that the iodine solution is unable to penetrate through the leaf and the iodine just sits on top. The reason for that is that the leaf has a layer of wax on top of it, prevents anything going in, stops the leaf from drying out in extreme heat and things like that. So this is no good. If we pour this away, we can just put a little bit of water on top, and you see that the iodine solution has not penetrated the leaf whatsoever. Right. What do we do to overcome that? Well, if we take our leaf and we put it into some boiling water, then what will happen is that the water will remove the wax from off the leaf, and it will start softening the leaf and breaking open the cells so that we can actually get to the stores of starches within the leaf. So all we do is, first, safety glasses. We've got some water here that we've already pre-boiled. It might take a little bit more, but we're going to pop the leaf just straight into the water. I'm just going to do it a little bit carefully. Pop it in, and we're going to give it 2 or 3 minutes just to soften it up. Bunsen up to full power, pop it under, and then we're just going to wait for it to boil, and you see the leaf just slowly changing colour as it cooks inside there, as the wax comes off, and then we can start to do the starch test on this leaf. While that's going on the board, I've got a very simple 2D diagram of the equipment that we did. You should be able to, from your years of with us in science, be able to label this. So we've got the tripod sitting on top of something that you should be able to name, and then the beak on top. And we're just placing the leaf into the water. We're going to give it 2-3 minutes. It would be very good if in your books, if you copy this one down, make sure you use 1, a pencil, 2, a ruler. You know how much we hate it when you get that one wrong. So please, pencil and ruler, and then label the pieces missing off the diagram. Should be able to name these pieces of equipment that we use in the
Let me
lab.
just come round and show it.
Yes.
Clearer.
Nice and clear. There we go. So I've already labelled the Bunsen. Remember, we just use an arrow. So what is this piece of equipment? What is this piece of equipment? What's this? What's this? And then obviously the leaf sitting in the water. We'll add to this diagram as we go along. Now, as you're doing that, we'll just check on our leaf, and you can see that it is beginning from the sort of green leaf that we started off with. It's beginning to change colour. Just give it a little prod, careful of the hot water, and probably another minute or so, and that should start boiling, and the wax layer coming off our leaf. If you remember your cells from previous topics, you should be able to be thinking about what part of the cell that water would start to be softening up. So let us get access to the starch stores. About another minute or so. Finish up your diagrams if you haven't, and I'll do a little dance. Right, as you can see now, we've got our leaf, big change in colour from the sort of bright green we had, and the leaf is now boiling. If I was going to eat this, this would probably be nicely cooked by now. The wax layer coming off quite nicely, and the cell wall softening down as it's in the boiling water. What we're going to do at that point is just take the Bunsen back to safety, and we're just going to take it out of the way. Now, the next part of this requires us to actually start breaking into the cell to get the contents out. Again, at this point, if I try and do a test with the iodine solution, I'm going to get nowhere on this one. So we're going to do a water bath part of the experiment to break open the contents of the cell. To do that, you're going to use ethanol. And ethanol, very, very useful thing in the lab. We keep this one in the fridge so it's nice and cold. Look at that. Proper ice on that one, so it's lovely and cold. Problem with ethanol, apart from people who want to drink it, is that it's incredibly flammable. So the biggest safety thing that we do here is we're going to turn the Bunsen burner off so that there are no flames around. Now, we're going to take the leaf and we're going to give it a drink of ethanol. The ethanol will do a wonderful job in breaking open the remainder of the cell wall, moving through the cell wall and the cell membrane, and also removing the green pigment, the chlorophyll, from the leaf. It allows us to then detect any colour change much better. So a little bit of ethanol at the bottom of the tube, and then we're going to simply fish out our leaf, and we're going to pop it into the boiling tube. Pop it down, and there it is. Okay, the next thing to do to accelerate this process is we're going to warm the ethanol. Before we had our hot water down here, and we're just simply going to water bath this one. The water is above the boiling point of ethanol, which is down much lower at around just under 70 degrees, I believe. We're going to pop it into the water, and you may be able to see straight away there's a tiny little bit of green already coming out of the leaf as the ethanol is beginning to dissolve through, remove the chlorophyll and allow us access to the contents. And we're going to leave that in there for a minute or two as it does its stuff. At this point, you can then add the boiling tube to your diagram. So you're going to have the boiling tube sitting into the beaker and then into a water bath for safety. One common entrance question is about why we use the boiling tube in the water bath. Why don't we heat it why don't we heat it directly? And again, that is because of the fact that ethanol boils at a very low temperature is extremely flammable. Now this is actually perfect. It's exactly what I want to see. And you can see that the water around the outside is not boiling, but the ethanol inside is boiling. We've reached its boiling point. It's beginning to work nicely on the leaf. Green chlorophyll beginning to dissolve into the solution there, and the leaf will start going a little bit white. Another minute or so, and then we'll move on to the actual test for this. Right, so this has been in here in the ethanol bath for 2 or 3 minutes, and if I pull it out you can see that the solution now has turned a nice deep green colour, suggesting that the chlorophyll has come out of the leaf. What we can do now is to prise that out of the ethanol, so we're going to use some tweezers, tongs, and just reach down and pull the leaf out of the solution. There, nearly. Try and get the whole leaf this time. There it is. No. And there we go. So we have a very nice cooked leaf. Just going to pop that down on the tray, and we're going to pop our ethanol down there. It is a lovely green. It looks like a neutral pH, doesn't it? There we go. Pop that in there for later. Now, at this point, we can now go into the leaf. We can just open it up, and we can see, compared to the leaf that went in, we've got a slightly unhappier leaf that has lost its green colour. Let's just do a quick comparison. So this was our leaf beforehand, and this is our leaf now. We've cooked it in water and ethanol. The green chlorophyll has come out of solution. If you feel it, there's a sort of feeling of wax on top of this leaf. This one here, you can't feel the wax at all. So we would think at this point we've probably broken through into the leaf, broken through into the stores, and we can now proceed with the test with iodine solution. However, before we do that, we're just going to make sure there are no other parts of ethanol on top of the leaf, and we're just going to dunk it back in the water just to clean it, just to make sure there's no other ethanol on it. Nice and carefully give it a wash. Any excess chlorophyll also comes off, so that water turns green a little bit. And now let's have a look. Let's break open that leaf again. He says. Okay, so little bits of green still left, but here the chlorophyll has definitely come out. Now we're going to take iodine solution again with the dropper. We're just going to put 2 or 3 drops of iodine solution onto the leaf and observe if there is a colour change. Now, at the moment, not a huge amount, but we'll keep going at different parts of the leaf and going round. I'm going to have to soak this one a little bit more. So this one requires a few more drops. Okay, I'm going to stop there, and we're just going to have a look at what we've got. If you remember your starch tests with iodine solution, you know that the solution starts an orange colour, and a positive test for starch is it turns a bluey black colour. Looking at our leaf, it's hard to tell where we've got positive results, but down here there's definitely no presence of starch. We still have an orange colour, no particular change. Up here there is a darkening. There's some chlorophyll left in these parts as well, but there is a small change from the orange to the blacky colour within the leaf. Now, one of the reasons this isn't a massively positive test is something to do with the season that we might be in. So our wonderful little plant here that has given up its leaves for us also knows that it's the height of winter. If we repeat this experiment maybe later in the spring, 3 months' time, 4 months' time, we would expect a plant to be photosynthesizing much stronger, much more light around higher temperatures, and therefore we would expect a greater store in this leaf of starch. So not a great surprise that this leaf has not been photosynthesizing a lot. Small amounts of stores to see it through the winter, not a huge amount of photosynthesizing. So absolutely, at this point of the year, we'd say that was pretty good and backs up our understanding of photosynthesis.
Thank you, Mr. Kibax. So perfect. So you've now seen how to test a leaf successfully for starch. And although it's in the middle of the winter, it's currently, when I think we filmed this, it was the 5th of January. It was a very cold day. I'm not at all surprised that the plant wasn't photosynthesizing like mad. Oh, it's a tongue twister. So what I've got for you now is a task. What I want you to do is these following things. I would like you to write down the stages of testing a leaf for starch. Now, you can always rewind the video and go back over them if you've forgotten, but I'd like a diagram for each of those stages as well. Don't forget ruler, don't forget pencil. Or if you're doing it on OneNote, then you can use the straight lines using the drawing application. At the bottom of that, I've got some questions that I really would like you to answer. So here are the questions. Why does the iodine run off the leaf? So when you first put the iodine on, it ran off. You need to be able to explain what that is. What does boiling do, in this case, to the leaf? What does the boiling do to the leaf? Why do we boil it? What is the purpose of that? Question number 3, is chlorophyll soluble in water and ethanol? Now, the clues were there in the explanation. Which one do you think chlorophyll is soluble in and why? Give a safety precaution needed for the experiment. Mr. Kibax was very specific about the safety precaution needed for this experiment. And the last of which, what is the tested result of the iodine test? So we did see some positive signs. What were those positive signs? So that's all very good. We've proven that the leaf contains starch. How can I then use this to measure biomass? Well, it's simple. You take your organism, in this case a plant, because we're measuring photosynthesis, and we weigh it at the start, and then we weigh it at the end. We check the mass. Now, we should technically do this when it's dry, because the amount of water is going to affect the weight, and the amount of water isn't part of the biomass. It's not a living weight. It's just part of what goes in and out of the organism. So we should dry it before and after, but if we regularly water it and keep it at the same, you can still show it that way. So you grow a plant, and as it grows, it gains mass. The quicker it photosynthesizes, the quicker it will gain mass. Simple. So let's recap. Here is our summary. We can measure rate of photosynthesis using 2 methods. We can use the bubble method, which involves using a plant underwater. That's more precise, that's quite easy to do, except for that it can only be done with plants that are aquatic, and that obviously presents problems because a lot of plants are not aquatic. Or you can use the biomass calculation. The problem with the biomass calculation is you have to dry it out, and that tends to kill the organism. So those are our 2 methods that we can use. Also, and it's a really common question, they ask you to describe the stages for testing a leaf for starch. You need to be able to name all of the stages, describe what's happening, explain why, and give safety precautions. Thank you very much for listening. I'll see you again soon. Bye-bye.